You know, one of the biggest questions out there is, how did life even start? Before there were cells or any kind of creature, there was just this chemical chaos. So how did that primordial mess organize itself into the very first spark of life? Well, today we're going to dive into a super cool clue, a potential ancestor found inside a simple little droplet.
So here's the deal. It was all about containment. For the machinery of life to get going, all the important bits and pieces had to be kept together. They needed some kind of room, a container, a wall, to keep them separate from the rest of the world. Without that, everything would just float away. But how do you build a wall when you don't even have a blueprint?
And that, right there, is the heart of the problem. It's what scientists call the concentration problem. Just imagine the early Earth. You've got this gigantic primordial soup, basically a planet-sized ocean, full of all the building blocks for life. Molecules like RNA, which was probably the precursor to DNA. See, for any of the magic to happen, you gotta bring those molecules together in one spot. They need to be concentrated so they can start bumping into each other, reacting, and building more complex stuff. Now, today, life uses these beautiful, complex, fatty cell membranes to do the job. But 4 billion years ago, nope, those didn't exist yet. That first container had to be much, much simpler. So what in the world could have been that first simple container? Well, researchers digging into this question have stumbled upon a really elegant and surprisingly simple answer. Let's meet the hero of our story. The whole thing starts with these simple molecules called α-hydroxy acids, or AHAs. Now, don't worry about the long name. What's really cool is what they can do. They were all over the place on the early Earth. We even find them in meteorites that fall to Earth today. And they have this neat trick. They can link together really easily and form long chains, creating a material that's a lot like polyester. So how do you make this happen? Well, it's kind of like a primordial baking recipe, and it's super simple. You just take these AHAs, dissolve them in water, and then let the water evaporate, like a puddle drying out in the sun. That process forces them to link up. And then, when you add the water back, something pretty amazing happens.
Boom, they just spontaneously form these incredible little microdroplets, which you can see right here. And that is the solution in action. These aren't fancy cells with complicated walls. They're just simple bubbles made of polyester chains. But you know what? They work. They create these tiny, self-contained environments, a little room to gather up other molecules and solve that massive primordial problem.
Okay, so we've got our container. That's a huge win. But a box is only really useful if you can put stuff inside it, right? So the next big challenge was to see if this primitive container could be upgraded to do something really useful, like interact with the most important molecule of them all, the code of life itself. And this is the million-dollar question, right? Could these little polyester droplets be tweaked so they don't just exist, but they actively pull in and hold on to RNA? Because if they could do that, it would be a massive step toward a real, functioning protocell. To figure this out, the researchers added a special ingredient to the mix. All right, check this out. On the left, you've got our standard building block. But on the right, that's the new addition. The key difference, and you can see it right there, is that little piece sticking off the side. That's an amino group. And that tiny change makes all the difference in the world. It's really just basic physics. RNA, as a molecule, carries a negative electrical charge. That little amino group on our special new ingredient, it carries a positive charge. And, as we all know, opposites attract. The whole idea was that this positive charge would act like a tiny magnet, pulling that negatively charged RNA right into the droplet. But of course, it's never as simple as just tossing it in the mix, right? Life is all about balance. The researchers had to figure out the absolute perfect chemical recipe to make a droplet that was both stable and attractive to RNA. And this is where it gets tricky. It's a real balancing act. Adding a new function is awesome, but not if it breaks the original structure. If you add too much of this new charged ingredient, the polyesters get too friendly with water, and they just dissolve instead of forming those nice little droplets. The recipe has to be just right. So, what did they do? They tested a whole bunch of different recipes. Think of this chart here like a map of all their experiments. Each column is a different mix of the ingredients, and as you go down, they're adding more and more of that special positively charged molecule. The black areas are the treasure. That's the sweet spot where you get perfect little droplets. The white areas, total failures. And here's what that looks like in real life. On the left, we've got a good recipe with just the right amount of the special ingredient. And you can see this whole field of beautiful, stable droplets. But now look at the right. They upped the special ingredient way too much, and nothing. The recipe failed. No droplets. It all just dissolved. It just goes to show you how critical that chemical balance really is.
All right, so they've nailed the recipe. They can reliably create stable droplets that have that special positively charged ingredient mixed right in. The stage is set for the big test.
So, does the theory actually work? Is that tiny electrostatic magnet really going to pull RNA inside these little primordial containers?
And look at that, it worked. It's a total success. In this image, the RNA was tagged with a red fluorescent dye so we could see it, and it's as clear as day. The RNA isn't just floating around anymore. It's been grabbed and pulled inside, and it's highly concentrated in those droplets. I mean, this is a massive deal. A simple, non-living chemical bubble made with a totally plausible primordial recipe just gained the ability to grab the code of life.
But wait, the story isn't even over yet. You know, like a lot of great moments in science, when the researchers looked a little closer, they found something they weren't expecting at all. A totally unexpected and really beautiful surprise. Turns out, their new and improved recipe came with a secret bonus feature, a hidden property that just kind of emerged from this new combination of ingredients. Get this, the new droplets were actually glowing, all by themselves. They were fluorescent. And not just one color. Look at these rows. When they looked at the same droplets under different filters, they were emitting blue, green, and red light, all without adding any kind of dye. This was a brand new property that just appeared out of the new chemical mix. And this, my friends, is a textbook example of what scientists call an emergent property. It's a new, complex behavior, in this case glowing, that you don't find in any of the individual ingredients by themselves. It only shows up when you combine them in just the right way. It's the system becoming more than the sum of its parts. And that just leaves you wondering, right? This whole story, from a simple bubble to something that can grab RNA to something that glows, all of that happened just by slightly tweaking a simple chemical recipe. If a tiny change like that can create totally new functions out of thin air, what other incredible secrets to life's origins are just hiding in plain sight, waiting for us to mix up the right recipe?